Hello lovelies, in this video Lauren, my expert examiner, is going to be taking you through AQA GCSE Biology topic 2, that is organisation. So what we want you to do is to make careful notes on each of this, pause the video, make notes and then watch it, make sure you've got it correct. But if you want more help with this, then over on the website there's a set of free questions, a set of free flashcards to help you revise, as well as the predicted papers and details of all the live and written sessions that we're running.
We need to know how living organisms are organised, so from the smallest part all the way up to the whole organism. Cells are the smallest living unit and the most basic building blocks of life. A group of similar cells working together to carry out a function is known as a tissue. A group of tissues working together to carry out a function is known as an organ. Then groups of organs come together to form organ systems. Multiple organ systems work together to form the whole organism. So an example of a tissue is a palisade tissue layer in a leaf, which carries out photosynthesis. An example of an organ is the heart, which pumps blood around the body. An example of an organised system of organs is the nervous system. And then a whole organism would be something like a whole plant, a whole insect, or obviously a human. And so we need to be able to understand that these go in size order as well. So cells are the smallest, then tissues, then organs, then organ systems, then the whole organism. Here's the diagram of the labelled digestive system. You should hopefully know this from Key Stage 3 and be able to label one of these exactly like this yourself. The salivary glands in the mouth secrete saliva that contains the enzyme amylase. This digests carbohydrates. So an example is starch, which would be the substrate. The enzyme is amylase, and they react together and it breaks down the starch into glucose, which is smaller sugars. The stomach contains hydrochloric acid to kill bacteria and secrete protease enzymes to digest proteins. So this is where we have proteins. The protease enzyme breaks these proteins down into the smaller molecules, which is amino acids. The small intestine is where the absorption of all the small molecules into the blood occurs. It contains all three types of enzymes that are secreted by the pancreas, including lipase to digest lipids. So lipids are broken down by lipase enzymes into glycerol and fatty acids. So on this page, we need to be able to remember all of the enzymes, what they break down, what they break their substrate down into, so the product or the small molecule. We need to know where they are secreted from, which I've written down here, and where they work. So in the salivary gland is where amylase is made and it works in the mouth. Protease is made in the stomach and it works in the stomach. And then the pancreas makes all three enzymes, a form of amylase, proteases, and lipase. So it's made in the pancreas, but they work in the small intestine to complete digestion, and that's then where all of those small molecules are absorbed. When we're talking about enzymes and how they work, we need to understand that they work through this idea of the lock and key mechanism, where the substrate acts like a key and the active site of the enzyme acts like a lock. It has a specific shape so that only one substrate can bind to the enzyme. Digestive enzymes are really important because they break down large insoluble molecules to small soluble molecules which can be absorbed through the cell membranes and into the blood to be transported and built into new molecules or for respiration in all the cells in the body. Amino acids, the small molecules that are absorbed, can be used to build proteins again for growth. Glucose can be used to build other carbohydrates like glycogen as an energy store, or they can be used directly in respiration to release energy for cells. The fatty acids and glycerol can be used again to make lipids, which can act as an energy store, or they can act as insulation. The rate of enzyme action, or the rate of enzyme-controlled reactions, can be affected by a few things. One of them is temperature. If you increase the temperature, you'll increase the rate of reaction, unless it gets too high, and then the enzyme will become denatured. pH is also a factor that affects enzymes. Outside of the optimum pH for an enzyme, it can denature and therefore slow down the rate. And surface area of a substrate, or the concentration of a substrate, if you increase this, it will increase the rate of reaction because there's more substrate and more surface area for the enzyme to bind to. When we say denatured, what we mean is the active site changes shape and therefore it can no longer bind to the substrate. The last part of digestion we need to know is about bile. Bile is produced by the liver and it neutralizes stomach acid in the small intestine. It also helps to digest fats through the process of emulsification. So it is made in the liver, it is stored in an organ called the gallbladder, which then releases it into the small intestine, which is where it works to help digest fats. Bile is not an enzyme. Increases the speed of digestion of lipids, but it does not actually do the breaking down. It is not an enzyme. It just binds to large lipid droplets, breaks them into smaller lipid droplets, which increases the surface area so that the lipase enzyme can bind to a greater surface area and therefore break down the lipids into glycerol and fatty acids faster. For all those molecules we talked about during digestion, we need to be able to show how we can identify these different molecules in food using various chemical tests. For starch, you would add iodine, and if starch is present, there'll be a colour change from yellow-brown to blue-black. You must say blue-black, or you can say black in the exam, but you cannot just say blue, because this is the colour of other test reagents. For proteins, you would add biuret reagent. The colour change, if protein is present, is from blue to purple. For glucose, you would add Benedict's reagent. We must spell this with a capital letter, same as biuret, because they are named after people. And you must heat it up. It will not work at room temperature. You should say heat, boil, or specify it's over 80 degrees. If glucose is present, there'll be a colour change from light blue to brick red. For lipids, you would add ethanol, then add water, and shake to mix it together, and a cloudy white emulsion will form. A negative result for any of these tests would be no colour change. So staying yellow-brown for starch, staying light blue for protein and glucose, and then no emulsion present for lipids. There are many ways that you can measure the rate of an enzyme reaction, and you could be given lots of different examples in the exam. The independent variable could be pH or temperature or substrate concentration, because changing all of these can affect the rate of an enzyme reaction. Depending on what the reaction is and what you're measuring, it's going to change the dependent variable. So you could measure a colour change using an indicator, so the time it takes for a colour to change to indicate that a substrate has been used up. You could measure the mass or volume of the product of an enzyme reaction. You could measure a change in pH. So for example, when lipids break down, they produce fatty acids. So the pH will decrease as more fatty acids are produced over time. So you can measure this with a pH meter. There are also control variables for each experiment, but similar ones will be things like volumes of solutions, the concentrations of solutions, and then pH or temperature, whichever one you are not changing as your independent variable. The example you may have looked at is starch and amylase. In this example, our independent variable is temperature, and we could use a water bath to vary the temperature of the different reactions. Our control variables would be using the same concentration and volume of both the substrate and the enzyme. And then we can measure the time that it takes for iodine to stop changing blue-black to show that all the starch is digested. So taking small samples of the mixture solution every 30 seconds, testing it with iodine until it no longer turns blue-black to show that all of that starch has been digested by the amylase enzyme. This is a graph of something predictable, like a similar result that you might see, where we've got the temperature on the x-axis and the rate of reaction on the y-axis. We normally see this mountain shape, where we can describe that as the temperature increases, the rate of reaction increases due to the increased kinetic energy of the particles. There's a peak, which is the optimum temperature, and then after the peak, the rate of reaction decreases as temperature increases because the enzyme is denatured. You'll notice that most human body enzymes, like the ones we've been looking at for digestion, will have an optimum temperature around the same as human body temperature, which is about 37 degrees C. We need to know the 3 different types of blood vessels and how their structure relates to their function. Arteries transport blood away from the heart at high pressure. This is how I remember the A for arteries and A for away from the heart. They have a narrow lumen to maintain high blood pressure. They have a thick elastic layer so they can stretch and recoil to maintain high pressure. So the maintaining high pressure here is part of their function, and this is how it relates to that bit of structure. So if you're describing and explaining the structure and function of arteries, you need to talk about saying the narrow lumen is a structure. What does it do? How does it help it carry out its function? It helps to maintain high pressure. Lastly, it has a thick muscle layer to withstand high pressure. You need to use these words, otherwise you won't get marks. Veins transport blood to the heart at low pressure. They have a wider lumen to transport a greater volume of blood. They have a thinner muscle and elastic wall layer because their blood is being transported at lower pressure. So we do not need the same features we see in arteries. Veins have valves. I remember this with the V for vein and the V for valves. This is to prevent the backflow of blood due to gravity. Capillaries transport blood close to cells so that substances can be exchanged. They have a very narrow lumen to slow blood flow down to one blood cell at a time. This increases the time for diffusion to occur. They have thin walls that are only one cell thick to decrease the diffusion distance. And ultimately they join small arteries and veins together, so once exchange has happened, the blood flows into veins and then can return back to the heart. You need to be able to use all of this information to be able to compare the structure of the different blood vessels as well as their functions. Blood is a tissue which transports substances around the body and plays a role in the immune system. The majority of blood is made up of blood plasma, which is the liquid that blood cells are suspended in. It's mostly water, but it contains all the dissolved substances that are being transported around in the blood. Things like glucose and other things like antibodies, for example, and other dissolved substances from the digestive system, like the amino acids and things that need to travel to and from your cells. The rest of the blood is made up of 3 different types of blood cells that are suspended in this liquid. They're red blood cells, white blood cells, and platelets. Platelets are involved in clotting open wounds to try and prevent infection or to prevent excess bleeding. Red blood cells are specialized cells because they have no nucleus. They contain lots of the hemoglobin protein, which is the red-coloured protein that binds oxygen. They have a biconcave shape, which gives them a large surface area to increase diffusion of gases into and out of the cells, and their job is to transport oxygen to and from cells. There are 2 types of white blood cells: phagocytes, these engulf pathogens, and lymphocytes. These produce antibodies or antitoxins. Both of these defend the body from pathogens and prevent you from getting sick, so they play a part in the immune system. We need to be able to label and describe the structure of the heart. First of all, with any heart diagram, always write on the right and left, the right way round, so it is the opposite to how your hands are on the page. Then we won't get mixed up with our rights and our lefts. The two top chambers are the atrium, and the two bottom chambers are ventricles. Think about it as A comes before V in the alphabet, so A is at the top and V is at the bottom. The blood vessel that brings deoxygenated blood from the body to the heart is the vena cava. It is a vein. Once blood has entered the right atrium, it's going to flow down into the right ventricle, and the contraction of the right ventricle will push the blood up and out of the heart through the pulmonary artery. It is an artery because it's taking blood away from the heart, and it is taking blood to the lungs to be oxygenated. When blood returns to the heart from the lungs, it returns in the pulmonary vein. So because it has been to the lungs, it is now oxygenated blood. This is the only vein in the body that carries oxygenated blood. The blood then moves through the left atrium, which contracts and pushes it into the left ventricle, which will contract and push it up and out of the aorta. This is the biggest artery in the body, and it takes blood at the highest pressure because it has to send it to the whole of the rest of the body. The left ventricle has a thicker muscle wall. This is so that it can contract with greater force, so that blood is forced out through the aorta at higher pressure, because it has to go a further distance to get around the whole body than when it leaves the pulmonary artery and just goes to the lungs. The human circulatory system is known as a double system or a double pump system because the blood flows through the heart twice, once to the lungs and once to the rest of the body. So the right side sends blood from the body to the lungs, and the left side sends blood from the lungs to the rest of the body. The heart rate is controlled by a group of cells known as the pacemaker cells that are found in the right atrium. If these cells stop working, they can cause arrhythmia, which is an irregular heartbeat, and an artificial pacemaker can then be fitted to control and regulate the heartbeat. The right-hand side of the heart pumps blood to the lungs to be oxygenated and to remove carbon dioxide. You can see a diagram of the lungs here. We should know the trachea, or the windpipe, branches out into two bronchi, and then the alveoli at the end of the tiny branches, which are known as bronchioles, and we have two lungs, one on either side. Here we've got a diagram of one alveolus from a bunch of alveoli and the capillary that's next door. You can see both of the walls are only one cell thick, as we mentioned, as this is an adaptation. We have deoxygenated blood coming from the heart on the left-hand side. When it reaches the alveolus, then diffusion of carbon dioxide from the blood into the alveoli will happen, and then oxygen will diffuse from the alveoli into the blood. This is where it will bind to the red blood cells, which will carry it and transport it back to the heart. Once the oxygenated blood will arrive back at the heart, then the left-hand side of the heart will pump this to the rest of the body. Coronary heart disease. The coronary arteries are blood vessels which transport blood to the heart muscle itself. If these coronary arteries get blocked with a fatty buildup called plaque, then blood flow is reduced, so less oxygen and glucose can reach the heart muscle. Without enough oxygen and glucose, the heart muscle cannot respire, so it stops contracting. This can cause a heart attack. Some risk factors for coronary heart disease, that means they increase the risk or the chance of you getting coronary heart disease, are poor diet and lack of exercise. This is because they can lead to other conditions such as high blood pressure, high cholesterol, and obesity, which are all medical risk factors for coronary heart disease. We need to be able to describe some treatments for coronary heart disease. Statins are drugs that can be taken to lower cholesterol. These are a preventative measure, so they cannot reduce any buildup that is already there, but they can reduce future buildup of plaque or fat in the coronary arteries in the future. Stents are a metal mesh-like cage which is used to widen arteries that have been narrowed by this fatty buildup. This can help to increase blood flow. With serious coronary heart disease, it can cause heart failure. If heart failure has been caused, then the options for treatment are a heart transplant or a heart bypass, or an artificial heart, which can be used to keep someone alive until a transplant becomes available. You need to be able to discuss the advantages and disadvantages of these different types of treatments for coronary heart disease. Health is a state of physical and mental well-being. Causes of ill health include disease, which can either be communicable disease or non-communicable disease. Communicable diseases are diseases that can be passed from one person to another, and they are caused by a pathogen. Non-communicable diseases cannot be passed from one person to another. Diet, because this can lead to certain non-communicable diseases. And stress, which can be caused by social or lifestyle factors. Different types of diseases might interact with each other, and you need to know how. Viruses can infect cells and cause cancer. For example, HPV can cause cervical cancer. Immune reactions to infections with a pathogen can trigger allergic reactions like skin rashes and asthma. So asthma is a form of allergy. Severe physical ill health caused by disease can lead to depression and other mental illnesses. Non-communicable diseases cannot spread between organisms. They're not caused by pathogens. They are usually long-term and get worse slowly. Examples include asthma, diabetes, and heart disease. Risk factors can increase the chance of getting these diseases. Non-communicable diseases can be caused by genes or genetic factors. These are out of a person's control. They could be caused by a mutation, or they could be inherited from their parents. The following are all risk factors related to lifestyle and often choices. Alcohol has been linked to effect in unborn babies' brain development. Excessive alcohol consumption has also been linked to liver disease and brain damage. A high-fat diet can lead to obesity. Obesity is a risk factor for type 2 diabetes, and a high-fat diet can also lead to high cholesterol, which is a risk factor for heart disease. Smoking whilst pregnant can reduce the birth weight of unborn babies. Smoking has been directly linked to causing lung cancer and lung diseases, and it also increases the chance of heart disease. Ionizing radiation, this is a link to your physics specification. So a carcinogen that damages DNA in cells, so it's an example of that. Exposure to ionizing radiation has been linked to cancer. For example, UV ray exposure has been linked to an increase in skin cancer. So you've got this idea that ionizing radiation will damage DNA, and then that can lead to it causing cancer, which we're going to look at next. But most diseases will be caused by a mixture of different factors coming together. It's not always just one of these. Cancer is a non-communicable disease where uncontrollable cell division causes tumours to form. There is some damage to the cell, normally to the DNA. This alters the way the cell division process occurs. This leads to uncontrolled or unlimited cell division, which then forms a tumour. Some tumours can be classed as benign. These are slow-growing and non-cancerous because they are contained in a membrane and do not spread to other parts of the body or other tissues. Malignant tumours, though, are cancerous. They are fast growing and they can break off and spread to form secondary tumours in other parts of the body or other tissues by travelling through the bloodstream. You need to be able to define cancer as uncontrollable cell division and know about the two different types of tumours. Risk factors for cancer. Smoking introduces carcinogenic chemicals into the body through the tar that's in the cigarettes, and it can cause lung cancer, but others as well. Exposure to UV radiation, which we said is a form of ionizing radiation, has been linked to skin cancer. Obesity is a risk factor because we know that fat cells can signal other cells to divide. Viral infection, we've talked about the example with HPV that can cause cervical cancer. Some cancers can be caused by genetic factors, so either mutations in DNA or inherited genes that can increase your chance of getting a certain cancer. For example, the BRCA gene, which is known to increase your chance of getting breast cancer. A leaf is a plant organ that is made up of different tissue layers. One of the tissues is the xylem. This transports water and mineral ions. The palisade mesophyll is a tissue layer that contains cells that have lots of chloroplasts in order to absorb light for photosynthesis. It's where the majority of photosynthesis happens in a leaf. The upper epidermis is a transparent layer of tissue that lets light through to the palisade layer for photosynthesis. The spongy mesophyll tissue is an open tissue with lots of air spaces between the cells, creating a large surface area for gases to diffuse, and it's where the majority of gas exchange takes place. The lower epidermis contains the stomatal pores that allow gases and water vapour to enter and exit the leaf. The phloem is the tissue that transports sugars. The guard cells are specialised cells in the layer of the lower epidermis. They control the opening and closing of stomata. They must balance the need to open the pores for gas exchange with trying not to make sure that they're open too often or for too long to prevent too much water also being lost through transpiration through the pores. The roots, stem, and leaves form an organ system in plants which transport substances. The main method of transport here is transpiration, which is the flow of water from roots to the leaves and is known as the transpiration stream. The process of transpiration is where water vapour evaporates through the stomata. The xylem is a tissue made of specialised cells that are adapted for the transport of water and mineral ions. Water flows up one way from the roots to the leaves through the transpiration stream, through the xylem, which is found in the roots, the stem, and the leaves. They have thickened walls of lignin to help them cope with this transport function, and their cells have no end walls between them, which allows this continuous column of water. Phloem is a tissue that contains specialised cells that are adapted for transporting the products of photosynthesis, mostly sugars, but also amino acids, around the plant, from the leaves to everywhere else in the plant that might need them. This process is known as translocation. The main adaptation of phloem cells is that they are elongated and they have little holes or perforations in their end walls to allow substances to pass through freely. Transpiration is the evaporation of water vapour through the stomata of leaves. Transpiration, or the rate of transpiration, can be measured using a potometer. So you can see the diagram set up below. This is just an example. Different potometers can look different, but we need to understand that we have a plant shoot, which will have leaves and stomata that the water can evaporate from, and that is connected all the way with an unbroken column of water from the bottom of the stem through a capillary tube, which has an air bubble in it. That's the only air bubble though. And we can move the air bubble to the start of the scale with the tap. And the air bubble is next to a scale, so we can measure the distance that the air bubble moves as water is sucked up through the plant shoot. And we are relating the fact that if it's sucking up more water from the plant shoot at the bottom, that is because more water is being lost at the leaves. You need to be able to explain how different factors can affect the rate of transpiration. Air movement or wind, if this is increased, it will increase the rate of evaporation of water, so therefore increase the transpiration rate. Light intensity, if this is increased, more stomata will open, so therefore this increases the transpiration rate. Temperature, if this increases, then the rate of evaporation is increased. Humidity, this is the only one that does the opposite, and transpiration will decrease as humidity increases because it decreases the rate of evaporation.
Ouch! This is why in some videos I explain scratches.